Today we are going to learn in vitro cytotoxicity analysis. In today's lecture, we will be learning in vitro cytotoxicity analysis and classification of in vitro cytotoxicity analysis. We will be also learning trypan blue exclusion and MTT assay both theoretically as well as practically in today's lecture. Let us see what is in vitro cytotoxicity analysis. Here we will be using the cell based assay to screen collections of compounds to check whether the test molecules or nanomaterials are cytotoxic or biocompatible in nature. Regardless of what type of cell based assay we are using, it is important to determine how many viable cells remain at the end of the experiment. And the selection of assay is one of the important parameters. We have to select the right assay and we have to select the right cell lines according to our material. What are the advantages of this in vitro cytotoxicity analysis? It is fast and cost effective, and it is reliable and reproducible. And also, this will be useful for testing large and different sample at the same time. Also, more prediction probability is possible here. Based on endpoint measurements, these in vitro cytotoxicity analysis are classified into different categories. The first one is dye exclusion method. This method is based on exclusion of dye by the live cells. And here the dead cells have ruptured plasma membrane, due to which the dye will penetrate inside and stain the dead cells. That means the live cells don't take up the stain, whereas the dead cells will take up the stain and the dead cells will appear blue color. If you are using trypan blue, the dead cells will appear blue color. The next assay is colorimetric assay. This technique is measurement of biochemical marker to quantify cellular metabolic activity. When the reagents come into contact with viable cells, they produce a color reaction, and the color can be quantified using a spectrophotometer. Some of the example for this assay is MTT and MTS assay. What happens is, if the more cells are viable, that will be more metabolically active, and this will convert the particular chemical into more color product. If you are getting more color, it implies that the cells are more viable. More viable cells means color intensity will be high. The next assay is fluorometric assay. This fluorometric assay is an alternative to dye exclusion and colorimetric methods. This fluorometric cell viability method is based on non-specific cleavage of non-fluorescent chemicals such as fluorescein diacetate, and which will give fluorescence when cleaved by cellular esterase. When this dye enter into the cell, inside the cell, the cellular esterase will break this fluorescent diacetate and it will produce fluorescence. The fluorescence signal can be measured, and that can give the number of viable cells. If we have more viable cells, that means more fluorescence. And the next assay is luminometric assay. This is similar to the fluorometric assay, but here we will be measuring the bioluminescence. This bioluminescent test is based on the correlation between a bioluminescent reaction and the impact of tested substance. The impact might be increased cell proliferation or cell death. Here we will be using this luminometer to measure the bioluminescence. Let us see the trypan blue exclusion and MTT assay more in detail. The first one is trypan blue exclusion assay. The trypan blue is a large negatively charged molecule, and it is used to determine the number of viable cells or dead cells in a cell suspension. The principle of this assay is live cells have intact cell membrane, which in turn it excludes the dye. As I told earlier, the live cells have intact cell membrane, so it won't take up the dye, whereas in the case of dead cells, it has a ruptured cell membrane. It will take up the dye and the cells will become blue color. In this assay, it can be useful for adherent cells or non-adherent cells, and we can treat the cells with serial dilution of test substance. For example, if you are using nanomaterial, we can use different concentration of nanoparticles, and we can incubate the cells for different durations. It can be for 24 hours, 48 hours, or 72 hours. Depends on the your material's toxicity, or depends on the experiment. We have to incubate the cells. At the end of the experiment, you have to wash the cells and suspend it in the appropriate medium. Then the dye will be added to the cell suspension. Once you add the dye, the viable cells will have a clear cytoplasm, whereas the dead cells have blue colored cytoplasm. And the number of viable or dead cells can be calculated by counting the number of blue color cells with the help of hemocytometer, or you can use the automated cell counter. Let us see the overview of trypan blue exclusion assay. Once the cells are 70% confluent in the tissue culture flask, remove the media and wash the cells with DPBS, that is dialbeco phosphate buffered saline. Then add the dissociating agent, that is 0.25% trypsin EDTA. Then incubate the cells in 5% CO2 incubator until complete detachment of cells. You can check the cells under the microscope to confirm all the cells are detached. Once the cells are detached, add media to the volume of 2x times of the dissociating agent. Then transfer it to a centrifuge tube and centrifuge the cells. Once the cells are centrifuged, the pellet contains a cell and the supernatant contain the medium as well as the dissociating agent. We have to discard the supernatant very carefully without disturbing the cell pellet. Then the cell pellet can be resuspended in media. Then we have to add 0.4% of trypan blue staining solution in a ratio of 1:1. Once it is mixed, add the stained solution in a hemocytometer and the cells can be counted under the microscope. Or if we have an automated cell counter, we can take the cell counting chamber slide and add the stained solution and insert the slide in the automated cell counter. It will count the number of cells and it will give the complete data, how many cells are dead and how many cells are live. We have to calculate the percentage of cell viability by number of viable cells divided by total number of cells multiplied by 100. By this, we can calculate the number of live cells. The next assay for understanding cell viability is MTT assay. The MTT assay is one of the most widely used calorimetric assay for assessing the cytotoxicity or cell viability. This technique is mainly based on the mitochondrial enzyme, that is succinate dehydrogenase. This enzyme converts the water soluble yellow color dye, that is MTT, into insoluble purple formation crystal. This can be dissolved and absorbance can be measured at 570 nanometer and we can calculate the cell viability. If we have more color intensity, that means more cells are viable. Let us see the overview of MTT assay. We have to add the cells with media into the plate in an appropriate count. We have to count and add the suitable number of cells in 96 well plate. Once the cells are added, incubate the cells for 24 hours in 5% CO2 incubator. Once the cells are nicely attached and grown, you have to add the test material. The test material can be nanoparticle or it can be a biomaterial or any small molecules or compounds. Once you add the test material, incubate the cells along with the test material for 24 hours or more than 24 hours according to the requirement of the experiment. Then remove the media carefully and add MTT dye. Then again incubate for 4 hours in 5% CO2 incubator. Then remove the MTT dye, then add the lysis buffer to dissolve the formation crystal. Incubate until complete dissolution of formation crystals. Once it is completely dissolved, then record the reading using plate reader. And the absorbance can be measured at 570 nanometer. As I told earlier, if you are having high color intensity, that implies that more number of cells are viable. Tabulate the data and the cell viability can be calculated by using the formula A test by A control multiplied by 100. Here this A denotes absorbance. Then we can analyze the data. For example, in this case, the control which is 100% and the sample 1, you can see the cell viability is more than 80%. That means the sample 1 is biocompatible material. If the cell viability is more than 80%, the material is biocompatible in nature. Whereas you can see that sample 3, the cell viability is less, which is toxic in nature. So, based on this, we can easily understand the cytotoxicity of your material. Let us see the troubleshooting for MTT assay. The first problem is MTT reagent is blue-green. It may be due to the contamination with a reducing agent, or it may be due to bacterial contamination. Discard it and you can remove and use the new MTT aliquot, or it may be due to excessive exposure to light. These are light sensitive, so you have to store it in a dark at 4 degrees Celsius. And the next problem is replicates have different values. Whenever we do the experiments, we have to always do triplicates. And if you are having a different values, it may be due to inaccurate plating or pipetting. For example, in the 96 well plate, you are adding the cells. The cell number should be uniform to all the wells. If there is a pipetting error or if there is a plating error, that will give a inaccurate results. So that's why to overcome this, we have to use the right number of cells when we plate the cells, and we have to avoid the pipetting error. Make sure that pipette is working properly. And third problem is absorbance readings are too high. So it may be due to cell number per well is too high. If the cell number is more in the wells, then the absorption will be very high. Then you have to decrease the cell density at plating, or it may be due to the contamination of culture with bacteria. The bacteria grows very rapidly. So if there is a bacterial contamination, that will also leads to increase in the absorbance reading. In this case, we have to discard it and we have to use the fresh MTT reagent and we have to repeat the experiment. And the last problem is the blank. If the only medium also is giving absorbance reading, okay, in that case the medium is contaminated with bacteria or this medium contains ascorbic acid. If it is contaminated with bacteria, we have to discard it, and if the medium is having ascorbic acid, or if you are not incubating this MTT assay in the dark, so that will lead to increase the absorbance in the blank also. So always incubate the plate in the dark, or we have to use the suitable medium, depends on the your cell type. And next problem is absorbance readings are too low. So it may be due to the cell number per well is too low, or the incubation time for reduction of MTT is too short. In that case, you can increase the incubation time until the purple color is developed. If the cell number is too low, we can increase the cell density while plating, and it may be due to incubation time for solubilization of formosant dye may be too short. In this case, we can increase the incubation time with the detergent reagent until all the formosant crystals are dissolved. And this may be also due to cells are not proliferating due to improper culture conditions or inadequate time to recovery after plating. That's what I told you in the MTT also. First step, once you add the cells to these 96 well plate, we have to allow the cells to attach and grow at least for 24 hours. Once the cells are nicely attached and grow, then only we have to add the test compound. So check the culture conditions and medium temperature and humidity, and make sure that the cell condition is proper before we add the test compound. Let us go to lab and learn this more in detail. To perform the cell culture experiment, we first need to UV sterilize the biosafety cabinet. Simultaneously preheat the required materials to 37 degrees Celsius. Once UV sterilization and preheating are complete, we will begin the cell culture experiment. For performing the experiment, we require DPBS, that is Delbecque's phosphate buffered saline, 0.25% trypsin EDTA, complete cell culture medium, 96 well plates, centrifuge tubes, micro tips, 3 sets of micropipettes, micro centrifuge tubes, glass beaker to discard the waste. Next, we will observe the T25 flask under the inverted microscope to check the cell confluency. If the cells are about 70% confluent, we can proceed with the experiment. Take out the flask from the CO2 incubator. Now we will examine it under the inverted microscope to confirm the confluency of the cells. Here you can see the cells are spread throughout the T25 flask, so we will proceed further. The first step is to discard the media and wash the flask twice with DPBS. After the washing step, we will add 500 microliter of the dissociating agent, that is 0.25% trypsin EDTA, and gently spread it throughout the flask. Now we will incubate the flask for 5 minutes in a 5% CO2 incubator. We will then observe these cells under the inverted microscope. As seen here, the cells have detached from the flask and we can now proceed to the centrifugation process. Next, we will add twice the volume of complete media to that of trypsin to the flask and slowly spread the media throughout the flask. Then transfer the entire mixture into a microcentrifuge tube. add an equal amount of media to a blank microcentrifuge tube. Now centrifuge both microcentrifuge tubes. After centrifugation, we can observe the pellet formed at the bottom of the tube. We have to carefully discard the trypsin containing media without disturbing the pellet. Next, we will add fresh media to the microcentrifuge tube. Using the media, slowly resuspend and dispense the pellet. Now we will add an equal volume of 0.4% trypan blue to the media containing cells and mix well. Next, add 10 microliter of the mixture onto both sides of the cell counting chamber slide. Switch on the automated cell counter and place the slide into the machine for cell counting. And press count cells. Once the count is complete, save the data to an external disk. And open it on the monitor to view the exact cell count. So from this data, you can clearly understand the percentage of live cells and dead cells. Here the live cells are represented in the green and dead cells are represented in the red color by the software. So here the live cells are 95% and the dead cells are 5%. Here you can see there are approximately 67 lakhs live cells present per ml. For the MTT assay and apoptotic studies, we need a concentration of 10,000 cells per 100 microliter. So for this, we will add 150 microliter of the cell suspension to 9.85 ml of complete media, which will give us the required cell concentration. First, add 150 microliter of the media containing cells to the centrifuge tube. Then add 850 microliter of media followed by 9 ml of media. Once we have prepared the exact cell concentration, we will seed the cells in a 96 well plate. One plate for the MTT assay and another one for the apoptotic studies. For the MTT assay, we need 6 wells, 3 for the control group and 3 for the test group. For the apoptotic study, we will perform 2 assays, AOEB and Hochst staining. So for this, we require 12 wells in total. Now we will add 100 microliter of media containing 10,000 cells to each of the 18 wells for both experiments. First, we will seed the cells for the MTT assay, followed by we will seed the cells for the apoptotic study. Here we have added the cells into 96 well plate. Now we will observe the cells under the microscope, then incubate the plate in 5% CO2 incubator to proceed with the experiments. We have seeded 10,000 cells for the MTT assay. Now we are going to view them under the inverted microscope to confirm whether the cells are present in both the control well and the well where the test material will be added. After confirmation, we will place the 96 well plate in a 5% CO2 incubator for 24 hours. Yesterday we seeded the cells for the assay. Today we will prepare the desired concentration of the test material for treatment. The concentration which we prepare must be diluted in the cell culture media. Once it is done, we will take the seeded cells from the incubator and treat them with the test material. Here we can see that the cells are well adhered to the 96 well plate. After confirming the attachment of the cells, we will treat them with the test material. For the control group, we will add 100 microliter of the cell culture medium, while in the remaining 3 wells, we will add 100 microliter of the desired concentration of the test material. We have to incubate it according to our treatment period. Here we will incubate it for 24 hours and we will observe the results after this incubation period. After the incubation, we have to carefully remove the medium from the wells and add 100 microliter of MTT dye. Since MTT dye is sensitive towards light, it should be added in a dark environment. Once we add MTT dye, we will incubate the plate for 3 to 4 hours. After incubation, we can observe the formation of Formazan crystals in the control wells, while the test wells show fewer Formazan crystals compared to the control. Next, we will carefully discard the MTT dye and add 100 microliter of lysis buffer to each well to dissolve the Formazan crystals. The plate will be incubated at 37 degrees Celsius for 10 minutes. After incubation, we observe the violet colored Formazan crystals. Finally, we will measure the observance at 570 nanometer and plot a graph based on its observance value. Here we have used IC50 concentration of a drug and studied its efficiency against the MG63 cell line. So that is the bone cancer cell line. And here you can observe the cell viability has gone down approximately 50%. So this indicates the IC50 concentration. So that means the concentration required to inhibit 50% of the cell growth. As a summary, in today's lecture, we learnt about in vitro cytotoxicity analysis and its classification. We also learnt trypan blue staining and MTT assay both theoretically as well as practically in detail. Thank you all for your kind attention. I'll see you in another interesting lecture.